11.3 Acquired Immunity. This is a slide we looked at in the last segment when we were distinguishing between innate immunity and acquired immunity. We defined acquired immunity as that is activated by exposure to a specific antigen. This is also sometimes called specific immunity because it is a response to a specific antigen. So we need to define an antigen. If you recall back in a previous lesson, we looked at the plasma membrane of the cell, and the plasma membrane is the phospholipid bilayer along with a number of embedded proteins and then various glycoproteins and glycolipids on the outside. Our immune cells recognize our own plasma membrane, our own cells and our own tissues. Anything else, however, is foreign, and this can initiate an immune response. So anything that is foreign, that is not part of our own cells or our own tissues, can be called an antigen. The two types of cells that are involved in acquired specific immunity are lymphocytes, B lymphocytes and T lymphocytes, and both of these have specific protein receptors in their own plasma membrane, and these recognize and bind to only specific antigens. So an antigen can be really anything that is not part of our own body. It could be a bacteria or even just a part of a bacteria. It can be a fragment of a foreign cell. It can be an infected cell, perhaps a cell that is infected with a virus. It could be something that we have ingested or injected, some foreign toxin molecule. So anything that is not of our own body can be an antigen, and a B cell or T cell can have a receptor that binds to that. The first type of specific acquired immunity that we'll look at is the antibody-mediated immunity. This involves the B lymphocytes. So it goes through several steps. If an antigen is present, a specific antigen will bind to a specific receptor on a specific B lymphocyte, and so we would say that this B lymphocyte has been activated, and that B lymphocyte then goes through some mitosis and produces cells called plasma cells. These plasma cells secrete antibodies, which are proteins, but the neat trick is these antibodies have the exact receptor specificity as the original receptor that was activated on the B cell. However, rather than being embedded in the plasma membrane of the B cell, these antibodies are going to be secreted. And so then they can be transported all throughout blood and tissue fluid or lymph, and they will bind to that exact same antigen. There are also some memory B cells that are produced. So this will allow a much quicker response in the event of a subsequent exposure. So here's a diagram that represents the B cell activation. This is called clonal expansion, but we begin up at the top with some B cells. They have a specific B cell receptor. If their antigen binds, they're activated. So they're in the middle of the diagram. You see the plasma cells are produced, and the plasma cells produce antibodies which are identical to the B cell receptor. And so they will also bind that same antigen. And then the memory B cells are produced again. So if we have a subsequent exposure, the response is going to be much quicker. Whenever the antibody binds to the antigen, that is called an agglutination reaction. And the agglutination recruits other components of the immune system, including phagocytes like macrophages that come gobble up these tagged antigens, and also it can activate the complement proteins in blood plasma. T lymphocytes are involved in cell-mediated immunity. Now there's some difference between T cells and B cells. They both have specific receptors. However, T cells are not able to recognize antigens without help. So what happens if a phagocyte, like a macrophage, engulfs some antigen, it will take a portion of that antigen and it will insert it into its own membrane, into the macrophage membrane, and then the macrophage will seek out a T lymphocyte and it will present the antigen. It's called antigen presentation. So what the macrophage is doing is finding a T cell, shows him the antigen, and basically says, here's a bad guy, now go find them and kill them. So this activated T cell divides to produce a cytotoxic T cell, and they are going to go on a search and destroy mission, and they are looking for anything that has the antigen that was presented to them. Whenever they encounter this, they will destroy that cell. So here is a diagram representing one of those cytotoxic T cells, and it has an antigen. It got that antigen from an antigen-presenting cell, but now it is going to go look for other cells that have that same antigen. If it finds them, there are some proteins called perforins that are inserted into the membrane of that target cell, and it will rupture and destroy the target cell.